Could I ask the panel to come and join me, please? Thank you. I think we'll start just to see if any of you have questions for each other or comments on each other's talks. I mean, there's a degree of overlap in some of the questions raised about collaboration here and the risks. There's been a common thread, I think, in some parts at least, on replicability of results. Any comments?
I don't have a comment on replicability, but in one of the comments you made about the sort of model that we've put together, you said obviously all the companies know all the results and therefore they're all going to choose the same targets. Amazingly, this actually hasn't happened. So that if we think we have something that they really should take up, you think all 6, let's say, would take it up, but we have every combination from 0 to 6 on every time this has happened. And even if several take it up, they often, target a different disease. Obviously, in our consortium, I mean, what the companies have agreed to do is not compete at the level of identifying the target, but they then compete at the level of the person who gets the best compound and chooses the right, therapeutic area to use the drug on wins at the end of the day. So we're just helping them at the very early stages of drug discovery, but surprisingly, the obvious doesn't happen.
That's very good news.
Any comments? No. So let's open up to questions or comments from you. Yes, please. I think there's a roving microphone coming through to you. If you could say who you are and where you're from, please.
My name's Jenny Laird. I'm from Eli Lilly. I wanted to ask a question about competition, not the kind that we usually think of between pharma companies or things, but more, I mean, a couple of the speakers mentioned this concept of the expert centre, or, the expert, usually academic and partly at least publicly funded. So what are your, various views of having only one? Can there be too many? How does it range, the competition between people doing essentially the same things, or potentially doing very similar, making a similar offer to the European researchers or global researchers? How do you see that
question,
spectrum?
I like that. So who would like to go first?
Shall I have a go? So I used to run drug discovery programs in my last job before I moved into target ID services. And so at one point we were interested in working on the kinase PDPK1, which there was several labs has got involved in that, both Dario Alessi at Dundee and also a guy at the Abraham Institute. So our interest in this was an adjacency one, because we saw PI3 kinase mutants in patients. We'd been working on AKT already, and so PDPK1 was a logical place to go. And so we needed some interpretation of the literature, because there already was some literature there. And so we were happy just to talk with one expert, who was Dario Alessi, and we didn't feel the need to ask the whole world about this, because, again, Alessi being a referee on lots of third-party papers, and obviously being in possession of lots of unpublished research as well, he had that informational quality we wanted to know. So we didn't need multiple experts. In a way, it's if for most drug discovery programs, you're coming in at a stage where a lot of basic information is already known, and so you have to accept that there's going to be competition for the discovery of particular templates. But and that's the choice you make. You could go for something really, really, really novel, but that comes with additional risk, which at Horizon we're trying to reduce.
Stuart, do you want to make a comment?
I think in terms of competition for public funds, that is quite an interesting issue. With respect to, there's been quite a rise in the number of academic drug discovery centres worldwide. Particularly in the UK, there's quite a lot now. And everyone's setting up the same shiny new infrastructure. So they're all very expensive systems. It costs a lot to, and then they're going out and employing similar people out of industry. And in that respect, it's a danger that funding is not necessarily going to basic sciences. So there has to be a very fine line walked, and I think it's very much driven by funding councils, because there has been a very significant emphasis on translational research. So everyone's kind of running around trying to set that up. So yeah, there is a danger. I think those who've got there first and set up the infrastructure are probably best set to service the community. But it's whether that collaboration, cross-institutional collaboration, can happen, really.
Philip, do you have a view on how many are too many, or whether one's enough?
How many companies or how many academic partners? Well, obviously, we find it convenient that all of our academics are in one place, because the companies just have to come to one place. You know, we're all there. Our students and postdocs can all participate, and so on. In terms of the competition, just in terms of competition between the companies for what we're offering. So, you know, even when we suggest things as a drug target, and some of the companies, you know, become interested and start to look at this more seriously, I should emphasize that none of the companies know what the other companies have chosen. You know, they don't know whether all 6 are working on it, or none, or one. As I say, we have all combinations, so we keep complete confidentiality. So no company knows what the others are doing, and you know, on occasion we sometimes have to do the same thing for several companies, or with different company compound. We don't know the structures of any of the compounds we're given, so but we may need to do similar things for several companies. But no company, you know, knows that, because we keep complete confidentiality.
So some sort of competition, I think, is good for innovation processes. And innovation doesn't normally happen unless there's some slack in the system to repeat things, to learn from them. I wonder, Jenny, if you wouldn't mind giving your view, seeing as you asked the question and you know quite a lot about this.
Well, I guess there are some, perhaps it varies depending on the topic or the field. I mean, certainly Sir Philip and the kinase collaborate. I mean, they kind of captured the market early, and they've collected a lot of expertise. And I think if I was going to leave Eli Lilly and, I don't know, get a job at Imperial and say, I'm going to set up a kinase center too in competition, it'd be tough. It'd be a tough sell. But I certainly completely agree with the comments of the others in the panel that maybe sometimes there are too many. Certainly we're seeing a lot of, you know, we're going to set up HDS in our university. And of course, all of the small, medium and large pharma have those facilities too. And they're slightly generic and expensive. And so there, I think, so I suppose I can summarize this by saying it's a balance. And maybe what each of the academic institutions or publicly funded institutions can really work on is what is their unique selling proposition. Because that I ask a lot of, you know, as you go around, you ask, and many people have one, but some people, it hasn't occurred to them. So.
Very good. Well, as an outsider to the world of drug discovery, it sounds to me as if this debate needs to be had in a healthy manner before we have too many and dilute activities elsewhere. And clearly, some competition. I'm giving the chair's balanced view here, aren't I? Let's move on. Who else has got a question, please, or a comment? Yes, sir. There's a microphone coming. No, it's right in front of you. Say who you are and where you're from.
Yes, so my name's Neil Martin. I'm from a company called Mission Therapeutics. This is for Sir Phil. I noticed that in the consortium it's all large pharma and it's all quite pricey. And for small companies like us who, you know, might enrich the pool a bit in terms of our thinking and our technologies and ideas, do you envisage a model where smaller companies could get in the mix? And be part of that type of consortium? We're
actually discussing this a lot at the moment, because no collaboration goes on forever. We're in our 18th year, so we're actually discussing with the current companies and with many other companies, you know, what they'd like to do with us in the future, and we're sort of throwing around all sorts of ideas. You know, it's possible that we could actually get vastly more money by having smaller collaborations with a much larger number of companies. So we're having an internal debate on all of this actually at the moment.
It's a great question, isn't it, for all of us? How do we collaborate with smaller companies, often those that don't have the machinery to, or the finance to make those collaborations work? How do these consortia pick partners?
Just make one other point, but actually, the model that we'd probably come up with is, you know, we might have a two-tier collaboration. You know, you might have a collaboration for companies who want to put a lot of money in and get everything out of it, probably companies who cover multiple therapeutic areas, but then you might want to have a consortium, let's say, of smaller companies who pay less, but they don't have the same sort of access to all of the services and reagents that are available, but only that get those that are actually in their very specific narrow area that they're focused on.
Rather than shopping at Matrix or shopping at Lidl.
Well, you still will get the same quality of service.
I mean, I think, back to the innovation question, it's a very important issue that fast growth smaller companies often are the bringers of new ideas into these networks. You know, look at biotech and so on. And how do we build models of collaboration where you are able to participate in a valuable way for both sides? And I think that's another takeaway from this discussion, that we need to find the right mechanisms. So hand up here, please.
Hi, my name's Cam Dalliwell from Horizon Discovery. My question is actually hopefully not too provocative, but I mean, what we're really talking about is open innovation, and this has been around for 20 years plus. I mean, I've been on these sorts of meetings, presentations, where people have spoken very passionately about the role of pharma, biotech, academia, CROs. So effectively, there is an inbuilt competition. We've had Jenny mention about how many academic centers should be doing HTS screening. You can see with the NIH, they've pulled funding, and a lot of those early roadmap initiatives, those universities who then built those structures, built the infrastructure, the compound collections, are now effectively CROs. So with pharma companies, you know, donating compound collections, as well as also phase 3 assets, what I would term fallen angels, to be able to reposition them in another indication. What do you think, going forward, for the good of the industry, should be the role of pharma, biotech, academia, and CROs? Because that's a different type of question, because you've seen the consolidation and changes in pharma, and, you know, where are we going to be in the next 10, 15 years? So that's the question.
Who's going to go first? Stuart? What do you think in 10 years' time we're going to be talking about in terms of the nexus of these different organisations working together?
Yeah. So I guess what the FPA companies, I guess, are hoping to get out of donating compounds to ELF is new chemical space for their own targets, but also a shopping window for new and interesting biology that might fit their portfolio, which maybe they'd not thought of before. So there does have to be a good route for basic academic science through to translation. And if it's deemed too risky by large pharma, somebody has to do it, otherwise they just sit on the shelf and they never get tested. So I think it is extremely important that there are mechanisms by which new targets can be tested for druggability, can go through models to validate or invalidate, and then hopefully provide that stepping stone through to some sort of product.
I actually think that what you're talking about is completely out with anyone on this panel's control. What will happen in the future is that everybody will do what they like, and everything under the sun will be tried, and the ones that are successful will win. But there's nothing you can do to stop anybody doing anything they like.
John, it was your colleague from Horizon Discovery who asked the question, I think.
What's my view on the role of different... I could be cheeky and ask to repeat the question because it's a very long one. But
I won't allow you to do that.
Thank you, David. There was, I think it's a question about what should the various roles be on the ecosystem. And, you know, much like animals or vegetables, you know, fighting for space, you know, on an island, one has to try and make a living as it goes on. So pharma, you know, they have their metrics, the research organizations such as they exist, you know, they have access to the same information, they have more resources. Pharma have the money, and so they can buy in programs at a later stage, which, you know, provides an opportunity for investors in biotech to get an access. CROs are best placed to build infrastructure on specialized services, whether they be commercial or in academia. So CROs really enable small commercial companies to compete on the scale by providing, you know, a properly built infrastructure which you only need every so often, and so it'd be very expensive to build bespoke. So I think those are the ways obvious things go. Maybe in the future there'll only be one drug company, which might mean there's less competition for the products of biotech, and it will all fall apart. But let's hope that doesn't happen.
Thank you. We've got time for at least a couple more. We're going to go to the gentleman up here first. Please say who you are and where you're from, and then we'll come across to you, sir.
So good morning, my name is Antonio Parada, and I'm the CEO of a biotech company that does antibodies for others. So my problem is the opposite. So how does research organization resist the temptation or not to do their own discovery programs and then start competing with the big pharmas that you're already collaborating with?
You've got a lot of experience.
I don't think you should stop it. Well, you can't do anything to stop it. I think, I can see some universities in the world turning from universities to pharmaceutical companies, because a lot of them have the capability of doing clinical trials and doing everything. So, you know, it hasn't, to my knowledge, happened quite like that yet, but it might do, particularly in private universities that are not, you know, accountable to public money.
Good. Any other views, colleagues? No?
No, I would agree with that.
Right. Thank you. Let's go across here.
I actually think this could be very important in the future, that let's say universities do turn into pharmaceutical companies. The problem at the moment is, of course, that all the pharmaceutical companies in the world are all converging on about 3 or 4 towns, creating a sort of huge hole for the translational work of the life scientists in other places. So for example, Scotland has, you know, 15% of the UK science budget, and it has a lot of great science and small biotechs, but you need a big pharma company to allow small biotech companies to thrive, because they have to sort of feed off of all of that. In part, it helps them, and it helps the pharma companies. So what does Scotland do to get a major pharmaceutical company going? Maybe a university, you know, with the wherewithal has to turn into a pharma company, and which would probably, you know, could be quite competitive in the future. Yeah.
Cheeky answer would be to talk to Nicola Sturgeon, but I'll leave you to do that when you're back in Scotland.
Well, I haven't proposed quite that, but I proposed something that eventually, due to a lot of infighting with Edinburgh and Glasgow, got downsized, and I got some money, but couldn't do quite what I was intending to do.
Good. Okay.
Hi, Kevin Cox from Imanova. I think we've seen some excellent examples of collaborations that help to de-risk target identification, target validation, but on John's very first slide, he illustrated actually where the biggest risk drug development actually is, and that's in the phase 2 proof of concept trials. What do you think we can do to form collaborations to reduce risk in that particular component of drug development?
John, do you want to go first?
Well, thank you for the hospital pass. So I think when it comes to phase 2, you know, someone's already decided 7 to 10 years ago to have a go at a particular target and the hypothesis. So really, you're coming up to that risk, and so your chances of success really are down to the decisions you made based on information that was available 8 to 10 years ago. I think one can, to de-risk something at that stage when you've already got an existing asset, really I suppose a company is going to get rewarded if it gets success in any of the potential indications where it might be used. And there may be companies which have got, maybe multiple companies which have similar assets at about the same time. So you could imagine some sort of consortia phase 2 trial where there was a pooling of risk and a pooling of resources. But I can't really see how the rewards are shared, because it's the different compounds will have different properties, and each clinical trial has to be done because you're testing safety as well as efficacy. So I'm not sure that the phase 2 transitions can be de-risked except by putting more resources in validating targets early on.
Stuart?
Well, yeah, I think the SGC model is trying to do just that thing. It's trying to get compounds through and validate or invalidate clinical compounds. So there is one model at least which is trying to do just that. Whether it'll be successful, as you say, it's one organisation who will be investing in one series presumably, and there may be liabilities that they don't predict, which competition might allow to get through if you had multiple people working at the same problem.
Philip, do you have a comment?
No, I don't have a comment.
Okay, fine. Well, look, we're getting very close. Oh, one last question. You've got your hand up in time, please.
I'm Joanne Rhodes. I'm Pfizer, but I'm actually embedded in Imperial College, working on translational science. And I'd just like to speak to that last question.
Please.
So one of the ways we've found of de-risking it, so I'm embedded and I'm looking at very early stage compounds, but in disease models using human primary tissue, based at the National Heart and Lung Institute. One of the things we found is we're looking very early, so de-risking targets. The main success we've had is actually we've stopped investigations really, really early. So we've thought, okay, this could be really useful in COPD. Let's test it in patient tissue immediately and have a look with a tool compound. And we've decided to actually exit that area very quickly without spending huge resources, developing primary screens and cell line screens and maybe testing in animal models. So the main benefit we've found is actually a cost saving, so we can then repurpose our drugs into other areas and other indications, and then we can refocus the money into looking at other assets in those diseases. So it's been very much cost saving rather than value added, but I think it's the same thing really, it's just two sides of the same coin.
Thank you.
But you can actually, invalidate things too quickly. I mean, there's actually, I've been told by actually quite several people that there's a lot of fantastic drugs that never sort of went far in phase 3 because the clinicians who did the trials were not very good, and that some wonderful opportunities were wasted. And I was talking, I'm not sure I should say this, but I was talking to Ron McKenzie, the head of R&D at Pfizer once, and I was telling him about, what a terrible waste that, there was this wonderful drug that should have been used to treat cancer that, failed, not because it was a bad drug or didn't have efficacy, but the way the clinical trial was done was no good. And he said, "Oh, that's nothing," and he proceeded to tell me about at least 5 other drugs that Pfizer had made that had a similar fate. But so the things can go wrong, even with drugs that are really good and way beyond, obviously, phase 2.
Thank you. Well, just to wrap up, I'd like to ask each of our panelists 2 questions, and I'd like quick-fire responses, just one sentence response, really. We've been talking about risks and collaboration. So what comes to mind quickly as the most significant risk, or the thing that would keep you awake at night about collaboration? John?
The risks about collaboration. The risk that's keeping you awake about drug discovery is investing in something that looks good and then not knowing you're fifth in the race and then getting no rewards for your effort unless it's substantially better than what's already. So that's the big unknown about what everyone else is doing, and I consider that the biggest risk. So it's a commercial one. So, and this is why you get this concentration in pipelines, because people are acting on info early.
Stuart?
I think the biggest risk with respect to what I do is a misunderstanding with the delivery of a compound or a hit list, in that people think that they then have a drug compound, and that there's a long way in the validation process, and that those compounds which may have spurious mechanisms of action or something, and we get misrepresented as producing poor quality chemical matter because of a lack of understanding of the process.
And Philip?
I mean, in our own collaboration, the main risk is that in a long collaboration like ours, that we don't evolve fast enough to give the companies enough, opportunities in the future. So, so their collaboration mustn't be, too static and rigid. It has to evolve. And my other risk would be that, we're really there to do, fundamental basic research and work out signaling networks that control biological processes, and out of that, comes new ideas for drug discovery, but we must be careful not to sort of do too much contract research, which we can sometimes are put under increasing pressure to do, but as someone else mentioned, in any collaboration, it only works if there's a little bit of give and take on both sides.
Thank you, Philip. And so just to close out, let's leave on an upbeat note. Just each of you, please give us a key ingredient for success in collaborative research. I mean, maybe, Philip, you've already said that through your comment on give and take. Do you have anything, other view on success?
Well, I've learnt an enormous amount from all of this, and I'm always looking for good ideas from pharma, and I've learned that pharma do things an awful lot better than academic researchers, particularly in the area of quality control. I've come to learn that a lot of academic quality control is absolutely terrible, and that's probably why a lot of the work is not reproducible. I can talk to people about this over lunch in more detail, but some horrendous things sort of come to light in the course of, we offer to do things in our cloning team for some of our academic colleagues as favors, and we find that it's a nightmare because of horrendous quality control that has a sort of a very long history. So I've learned an enormous amount from pharma and how to actually improve my own basic research. So we must be very humble and learn from our pharmaceutical colleagues.
Thank you, Philip. And John?
For a successful collaboration, the most important thing is to listen to your collaborator.
Great. And Stuart?
Yeah, just to elaborate on that, I think one of the benefits of collaborating with Dutch people is that they're very direct. and that you don't be afraid of being direct, and don't take offence when someone's being direct. So robust conversation is the best way to overcome a problem.
Absolutely. Thank you. Well, let's thank our panellists and have a robust conversation over lunch. Thank you.